Mr. Chairman, ladies and gentlemen, a warm thank you to Claudio Ronco for the invitation and for being such an inspiration over all the years. I'm talking about the CytoSorb system and what evidence we have today, one of the new kids on the block in adsorption technology, and this is mainly used in sepsis and septic shock today. And I review the data as I see it. Shortly for the pathophysiology, not really, well, a big need here for this audience, but as some of the inventors actually of sepsis are in the room, but this is work from Claudio, not from Claudio this time, but from John Callum, and just to resemble that our idea that highest mortality in sepsis typically goes along with high levels of cytokines, in this case IL-6 and IL-10. I think that was probably one of the ideas, and Claudio Ronco referred to the peak concentration hypothesis that he coined this term, as you know, that was leading to the idea that maybe if we remove these high cytokine and mediator levels, that this might impact the clinical course. Well, the CytoSorb, in fact, comes as a cytokine adsorbent, but what I think is, and we'll see this in a minute, that basically it probably can remove more from blood and plasma as just cytokines. What we have is a column that has some 300 cc's filled with polymer beads and saline, and the beads are binding unspecifically, but they have a size exclusion mechanism in a way that molecules larger than roughly 55 kilodalton will be excluded and most probably just go through. So this, and we saw this in a picture just in the last lecture, but this would open the room of the large inner surface to a number of very interesting molecules that now are discussed to play a role in the pathogenesis of sepsis, of course, so various bacterial toxins, but DAMPs as HMG B1, for instance, and of course the cytokines. By the way, myoglobin and free hemoglobin belong into that range, and there are groups studying the CytoSorb in this situation as well. Well, and as I said, larger molecules are somehow kept outside, like there's no much albumin adsorption, as albumin, as you know, is larger than that. It's not really fitting in. And of course, larger molecules as well as immunoglobulins or coagulation factors. And so in clinical observational trials, we do not really see these going down through this direct hemoperfusion device. How can we use it? Typically, I'm a nephrologist, we use it in combination with CRRT, and you can have it in a pre and in a post filter position. We typically have it like this in the pre-filter position, and but it can be applied with other extracorporeal techniques or on its own, just as a hemoperfusion. But there are others using this in the context of cardiopulmonary bypass surgery, as can be seen here, and similar combinations are possible in combination with ECMO. Well, this is the typical pictures. You could see it back home in Rostock, where I come from, and it's. I think we have it now for 5 years available, has a CE mark in Europe. It's a US invention, basically, but it started in Europe. And there is a registry that is steered by University of Jena, a hotspot of sepsis research in Germany. And everyone's invited to report their cases. And as of today, there are reports from 139 centers in the registry, and you'll see some data later on. Everyone would be invited, obviously, to report their data. Well, how did we start? We were among the first, I think, to use it in Germany, and our index case was this one, a case of an 80-year-old man, a dialysis patient, a stable dialysis, and he he fainted at the end of a dialysis session, and then when he woke up again, he had shortness of breath, had rales on both lungs, and had a fever of 39 degrees. So he was rushed to the hospital and diagnosed with sepsis, actually having a saturation of 79% and high scores. So he was brought to intensive care, intubated, ventilated. And then, as typically it is the case in Rostock, that nephrologists are called into to help with the cases when, well, colleagues are in need for renal replacement therapy. And this was the case, as you can see here. He didn't have kidney function anyway, but so we started him on CVHD, but we suggested to test for IL-6, what is in blue here, and there was some increase. It was not dramatically high, but much higher than the normal levels. So we suggested to take this new sorbent, on top of CVHD in pre-filter position, and then we let that run in combination for 24 hours. And we were rather pleased to see, yes, indeed, that IL-6 is going down. We do not know about other cytokines because that's not measured routinely in our hospital, so no information on that. What was clinically more important was that the colleagues could more or less decrease and shut down the norepinephrine dosage in that patient. So we were pleased with that case and said, yes, we would probably use it again. Well, what evidence do we have today? It's a new product, and so definitely work is on the way, and we have not answers to all questions. But first is, does it remove cytokines? And most evidence is for IL-6, as I said. And this is data from our centers, the first 9 cases we had put together. And yes, indeed, you can expect that your IL-6 levels in your patients go down. And this is a logarithmic scale, as you may see, and those that survived to 24 hours, there were 7 out of 9, 2 did not, had a more or less impressive decrease there. And others saw that as well. This is a younger publication of a series of 22 cases, and again, you see logarithmic scale that IL-6 nicely came down with this treatment in septic shock. And if we ask this registry that I introduced to you, then again we have the same signal that pre and post treatment we have a decrease from a mean of 5,000 picogram per milliliter to less than 300, what basically is adding to this evidence. Is there more cytokine removal? Probably yes. We have single reports from clinical tests. We have good evidence from John Callum's group from animal work. I show that in a second. But as I said, there's no routine measurement of other cytokines in our hospital. Clinically more important, obviously, is any impact on septic shock, the course of septic shock. And this is the same 9 patients you already saw, and 2 died on the way, and there was no improvement in norepinephrine dosage. But basically in all the others, we had a decrease after 24 hours. And that was most pronounced in those patients that started with the highest dosages. So that was somehow indirectly supporting Claudio Ronco's theory that those patients might take most advantage that have the highest levels of disturbing plasma compounds somehow. Mean arterial pressure was doing fine during that treatment, so there was basically going up in all those 24-hour survivors. So that was good. So we could bring a substantial number of our patients basically out of septic shock or improve it. And others found a similar thing following lactate concentrations. You see that this was increased in this cohort and nicely came down, and this was going along. These colleagues calculated lactate clearance that was negative and turned into positive during the course of this treatment. so indirectly giving sign of better organ perfusion and again, a nice idea to study in the future. Well, how does this work? Is this just an IL-6 sorbent? Yes, probably it is. But as you can imagine, it's direct hemoperfusion. It's an extremely large surface that is offered to blood. And fortunately, it's very well tolerated. And it can obviously bind more than just IL-6 or cytokines. And you saw the range of potential candidates there. One interesting, intriguing idea on how this might help, actually, among others with the hemodynamics, is this work that was focused on the integrity of the vascular endothelial wall. And we just heard about the importance of endothelial integrity in the last lecture. Here these were colleagues around Jan Kielstein, a colleague from Germany, and they had done treatments with a filter and then collected plasma and put that on human vascular endothelial cell cultures. And you see if they take a control patient, then you get a picture. If you stain for the intracellular glue, you know, there's cathedrin, and they had actin staining as well, then you get this neat picture of a solid endothelial cell layer. And the same culture with the pre-treatment septic shock patients gives a very different disrupted picture. And then you can figure that what they did is they had post-treatment plasma put on the culture, and it very much looked better than the pre-status. Well, that's an optical impression. What you can do on top of that is measure the integrity by doing the TEER test, or here it's the TER test. That's the trans-epithelial electrical resistance. What you want to have is a high resistance that will resemble a cell layer that's really tight. And see, here this is what they got with the treatment. Pre-treatment, they had a continuous loss of the resistance, and they could restore this and stabilize this with the plasma of the post-treatment, indicating improved barrier function. And they concluded that if there's any beneficial observations that might be reported from the treatment with the sorbent in septic shock patients, then this might at least in part be promoted via the protection of a vascular barrier function. I think it's a very intriguing idea, and we would very much like to study that in the future. Well, then, as this is obviously an interesting question, much too early to ask for definite data, but what information do we have on clinical course and outcome? Well, at least in septic rats, you know, they can take advantage from the treatment, as John Callum showed. And colleagues, what they did is have a cecal ligation and puncture model in rats, and they found various cytokines to nicely come down as compared to sham with TNF-alpha, IL-6, we saw that, IL-10, IL-1 beta, and they already could foresee this impact on hemodynamics with this more stable mean arterial pressure, and then most impressively, the very short-term follow-up, but there was this impressive difference in survival in rats, you know, so you can save animals. What human data do we have? No good control data yet, but there are some case series, and one is just published. A lot of the literature is from the last year, this year. These were 20 patients in what these colleagues call refractory septic shock. So everything was, the sepsis bundle was in place, but they had an ever-increasing lactate level or norepinephrine dosage need, more men than women. And you see the typical mixture, some 14 to 15 SOFA points, 25,000 IL-6 mean, and an increased procalcitonin. So those were treated. If they were not within 6 hours stable with their shock situation, they were declared refractory, and then started with the direct hemoperfusion. And from 20 patients, there was no shock reversal, early death, or a worsening of the shock situation in 7 patients. And in the other 13, there was a shock reversal, meaning that norepinephrine dosage really went down to near zero, and lactate levels were lower than 2.2 micromole per liter. Of those that reversed, there were 4 that did not survive to discharge, but 9 survived to 28 days. Sorry, there was 28 days survival. So we had 45% survival or 55% mortality. Well, what probably is not bad, so but definitely a situation that would be worth studying in the future if you compare what you do if you don't have a control group with what SOFA scores would tell us about statistical likeliness of mortality. Again, if we ask for survival data or mortality data, we could go to this registry, and again, there is a signal, that is hypothesis creating. With regard, this is the real reported data for ICU and hospital discharge survival as compared to Apache 2 and SAPS 2. It looks like, there might be a difference worth studying whether the treatment can, in fact, improve survival in the end. So I said we are typically called in as a nephrologist not within the first hours, something we are only now learning, with the emergency teams, but we don't have that yet in Rostock. So typically, if we come, then the kidney is gone, and we are asked for renal replacement therapy, and we then offer to combine this with the direct hemoperfusion. So we looked at our data with regard to timing, and so we had our first 30 patients, and we arbitrarily put a 24-hour timeline, after patients were admitted to ICU, and said how many were treated within the 24 hours or later, and we call this early and later treatment. And you see this was 50-50, roughly. And then we looked at whether we would see in Rostock some signal with regard to survival. And so we looked at those that we typically started with 1 or 2 columns, meaning 1 or 2 days of treatment. That's the typical mode, and we have no idea how many to use, actually. So we looked at those that survived to longer than 3 days, and those in the early, within 24-hour intervention group, we had some 60% survivors, short-term survivors, versus those that were started later were half of that. That was not significant and was not meant to do that as retrospective, but I think that's a clear signal that I would say we would always vote for starting the treatment within the first 24 hours. If we look at IL-6 scores in these 30 patients, then we see in the whole group, yes, there was a nice decrease, 77,000 to 30,000. That was highly significant. But then again, if we look at these 3-day survivors versus non-survivors, we saw that basically all the effect in decreasing IL-6 was traveling with this short-term survivor group. And there we had the very impressive decrease, but not with those that were started later. They were not only higher on average, but there was not this impressive effect. That was not significant. So this made us think that either we have a group of patients that we cannot save with this treatment, what is a fair choice and possibility, or we should do other protocols, more intensive treatments here, again, nice hypothesis to study in the future. Length of treatment, we looked at, typically, as I said, we'd order 1 column for 24 hours. In the whole group, we had, yes, basically 24 hours. Again, if we subdivide this to these short-term survivors, we saw that those were the ones having much more longer treatment, some 12 hours more treatment than those that were short-term survivors. Some probably died on the way, but still, if we had the feeling if we managed to keep a patient on the treatment and alive long enough to get some early start undisruptive treatment, then we see better removal of the markers and maybe better short-term survival, And this is pretty much in line with what another group found, again, a case series, 26 patients, and there was in critical care this year. And they saw a similar thing. if they group their patient to starting earlier than 24 hours or later, they saw that a score predicted mortality was higher than what they really observed, and this advantage disappeared somehow or was not that prominent if they started later on. And they concluded that the adsorption treatment resulted in rapid hemodynamic stabilization and increased survival, particularly in patients in whom therapy was started early. So again, there's no final proof, but I think it's, well, we are building up a story that even might end up in sufficient trials to prove impact on the clinical course. If you look at trials.gov, what I did this morning, you see, and you ask for CytoSorb, you'll find some 18 entries, so there's something going on. Many of those are recruiting or on the way to recruit and asking different questions around the sepsis story, obviously, but there's cardiac surgery as well somewhere around here, and there's acute pancreatitis and other indications, so we'll hear about that maybe in the future. And my summary, Mr. Chairman, is that we use it. Yeah, it's well tolerated. What is very nice for a direct hemoperfusion device, actually, in critically ill patients. It appears clinically safe. Hemocompatibility is very good. We can remove IL-6. Yes, we don't know whether that's the whole story. We think not, but we cannot really measure other stuff, or we didn't do. Treatments have a positive impact on hemodynamics, and that in a relatively short time. And there are signs of improved organ perfusion, if you want to read the lactate course as a sign for that. At least in Rostock, where I come from, it looks like early initiation and continuous intense treatment are correlated with better course and maybe better outcome. And there's tons of open questions, and we are happy and willing to study that in the future. Thank you very much.